A very good evening to all of you. We are the student enrolled in Rational Health Activities, the Faculty of Medicine, University of Colombo. I am Navin Chandika, who is moderating today's discussion. Today we have gathered to this discussion for a very timely manner as medical student. As we all know, we are in a very huge pandemic, which is predicted to go on for about another several decades. As audience, today's audience is mainly focused on the students who is from the first year to the last year, so for the final year. Regardless this batch who are working now, you might have at least one kind of this COVID-19 patient in your future goals. So, as medical professionals, it is our responsibility to have a sound knowledge about in and out of this disease to treat as best we can for our patient. So, for that, today we are focusing to have a knowledge how we can treat these patients and how vaccination works in Sri Lanka. So without further ado, let me introduce our resource persons today. First of all, today we have Professor Shalini Sri Ranganathan, the senior lecturer, Department of Pharmacology, Faculty of Medicine, University of Colombo. Good evening, madam. Then we have
good evening.
Good evening, madam. Then we have Professor Inoka Korea, Senior Lecturer, Department of Microbiology, Faculty of Medicine, University of Colombo. Good evening, madam.
Good evening.
Good evening, madam. And finally, we have Dr. Harsha Sathishchandra, Consultant Physician, National Hospitals of Sri Lanka. Good evening, sir.
Good evening to you all. Thank you.
So, before starting this webinar, I would like to have two announcements. First thing is, please keep your mics muted along this discussion. And if you have any doubts or questions to raise with from our resource person, you can leave those questions in the chat box so we can get those questions at the end of this discussion. So, first speaker of this webinar, I would like to invite Professor Inuka Korea to speak about COVID-19 vaccination. Nowadays, COVID-19 vaccination is no longer a strange topic to Sri Lanka, but there are still so many myths and beliefs about this vaccination rather than any other vaccine we have taken from our childhood. And also, as a commonly used vaccine, Sinopharm, we need to have a sound knowledge how this works and how it gives us immunity. And also, there are so many discussions about mix and matching several kind of COVID-19 vaccines. So, madam, can you give this some idea about how these vaccines work and how we can get those immunity?
Good evening, everyone. Let me share my screen first.
Okay.
So my video is not working very well. hope you can see my screen.
Yes,
Yes, Madam.
screen is seen, Inoka.
Thank you, everyone. So COVID-19 vaccination is a good way to start this webinar, because at the moment, it seems to be the only proven way to mitigate this pandemic until we come up with better treatment options. So prevention, like we always say, prevention is better than cure. And so therefore we are starting with prevention rather than with cure. So just as an introduction, since I'm the first speaker, remember, just to remind you that COVID-19 stands for Coronavirus Infectious Disease 2019. And we know it's caused by the SARS-CoV-2 virus, which again stands for Severe Acute Respiratory Syndrome Coronavirus 2, which belongs to the family of coronaviruses that, and you can see they have that crown-like appearance on electron microscopy. And why do they have that crown-like appearance? Because they have an outer lipid envelope with spike proteins. And of course, it is a positive sense RNA virus. So knowing all this, what are the vaccines that we could make for a virus like this? To prevent these dreadful things, complications, as you know, the virus, the spike protein, latches onto the ACE2 receptor and enters the cells of the body, especially the cells of the respiratory tract and the pneumocytes, and then hijacks the cells and turns them into virus factories. So the first thing to remember is, how does it enter? It is entering through the spike protein. So the most important sort of antigen that people have been targeting to use in the vaccines is this spike protein. And once you identify that your antigen is going to be spike protein, then you have to also think about what are the types of immunity you want to generate using your vaccine. So we know that viruses are viral antigens provoke formation of B cells, which produce antibodies that can block the virus when it is outside the cell in the viremic phase. But equally importantly, the viral antigens have to provoke the formation of T cells, especially cytotoxic T cells, which will go on to kill any virally infected cell. And of course, the T helper cells are required both for B cell antibody production and for the cytotoxic T cell function. So an effective vaccine will have to generate both arms of the immune response, humoral immunity and cell-mediated immunity. So there were many approaches that people were trying, which have been used previously for vaccines. Attenuated viruses, we don't have any vaccines at the moment, but inactivated vaccines we know about, and there are inactivated vaccines for SARS-CoV, such as Sinopharm and the Sinovac. Then there's a novel way of doing things. You could take a non-replicating virus, right? Say an adenovirus that is non-replicating. It has been sort of engineered in such a way that it does not replicate in the body. And you can then put in the SARS-CoV-2 genes into that virus, and so that it starts producing the viral proteins, coronavirus proteins, but it doesn't replicate, but it's continuing to produce these coronavirus proteins, and they will engender an immune response. So the vaccines you're familiar with, like AstraZeneca, uses that technology, and so does the Sputnik and the Johnson & Johnson. You could also use a fragment of protein to generate immune response, and Novavax falls into that category. You could use a DNA vaccine to that encodes the COVID-19 antigen, but we don't have any at the moment that are being used. And of course, we can have also the RNA, where we have a mRNA, just a segment of the viral genome that codes for our protein of interest. So here our protein of interest in all these vaccines, our protein of interest is the spike protein, right? So we have the RNA that codes for the spike protein, and we just inject the RNA, and we produce the vaccine in that way. So Pfizer and Moderna fall into that. And of course, the vaccines will also have adjuvants. Adjuvants are added things to stimulate the immune response, and also various packaging, things like, say, lipid nanoparticles or something like that. So the first few vaccines that came out belong to these different groups, as you know already, with the Pfizer and Moderna being RNA, and the vector vaccines, as I said, AstraZeneca, Sputnik, Johnson & Johnson, inactivated ones, Coronavac and Sinopharm, and now the Bharat Biotech has come out and seems to be really good. So these are now available, and most of them will be coming to Sri Lanka. So the mRNA vaccine, as I said, is just a messenger RNA in a lipid particle, and all that happens, it's
Can't hear you, Inoka.
You are muted.
Inoka, can't hear you.
Oh, did you not hear me at all?
Start with mRNA vaccine again.
Sorry, yes, I'm sorry. I think I accidentally muted. Right, so mRNA vaccines, as you know, mRNA, as you know, is just translated into the target protein, and it's just in the cell cytoplasm, and it does not integrate into your nucleus or anything like that, or into your genome, and will keep on producing the spike protein, and then you will generate your immune response. But the problem with these mRNA vaccines, as you know, have extremely low temperatures, like -70, -80. So that type of storage is very complicated. So here you can see how it works. The mRNA in a nanoparticle is released into the cell, and the spike protein is made continuously, generating the required T and B cells responses. Then we have the inactivated vector viruses, for like the AstraZeneca and the Johnson & Johnson. So there's a very sort of harmless virus. Usually we don't use human viruses because we'll have immune response to human viruses, so something like a chimpanzee virus. And then we put in the construct that we want, the DNA for the spike protein, into the adenovirus, because remember, adenoviruses are DNA viruses, okay? So it's the DNA now that is put into the virus, and then that adenovirus will then start producing this spike protein. So AstraZeneca, Johnson & Johnson, and also the Sputnik. The Sputnik is interesting because it uses 2 different adenoviruses for the first dose and the second dose, right? So it's a replication-incompetent vector. It has been engineered not to replicate, but to keep expressing this viral protein, right? And but of course, as I said, immunity to the adenovirus can attenuate the vaccine. So we use uncommon viruses like chimpanzee adenoviruses. Then, of course, we have the inactivated vaccines that you grow, COVID, you grow it in a cell culture and chemically inactivate the virus. And then you have to add adjuvants, because remember, inactivated vaccines usually have low efficacy. So you have to add some kind of adjuvant to make it work. But of course, the good side perhaps of an inactivated vaccine is that it will be not just the spike protein anymore. Now, remember, all the things we talked about earlier were just making spike protein, exclusively the spike protein. But here, this is an inactivated virus. And so all the components of the virus will be there. Right. So what we have to think about the vaccine. We have to worry about vaccine immunogenicity, vaccine efficacy, and vaccine effectiveness. So vaccine immunogenicity is just the measure of the immune response. So we can measure antibodies to the vaccine. Sometimes you can measure even T cell responses to the vaccine. And this is usually done in trials or in early trials to make sure that the vaccine is generating some kind of immune response. But the next stage, as you know, is to look for vaccine efficacy in clinical trials. And there you're really looking at, is there a reduction in disease incidence when you practically use the vaccine in people? So you divide people into vaccinated and controls, and then you look at the attack rate in unvaccinated and the attack rate in the vaccinated, and you do this calculation and you calculate the efficacy. So what is vaccine effectiveness? That happens next when the vaccine is approved after efficacy trials and it's sent out and it's used in the general population. It doesn't stop there. You keep on monitoring the vaccine and look for its effectiveness in the real life situation. Is it able to protect people in the real life condition? Remember, clinical trials are always done in highly chosen people. So we did not include pregnant women, we did not include children, we did not include this one and that one. So they were done in very selected populations. But so when you go into the real world and you start giving it to large numbers of people who are not specifically
Can't hear Inoka. She-
Madam, we can't hear you.
Inoka, we can't hear you. This.
I think you lost me for a moment.
Yeah, we lost you for 2 to 3 minutes, Inoka.
Okay, let me go back. I'm so sorry.
Slides also gone.
Yes. Chalini, can you tell me where it stopped?
Open the slides first. You went up to effectiveness.
Right.
Go to the slideshow. I will.
Yes.
In the
This
formula
one.
was there, efficacy formula was there.
Effectiveness.
Yeah.
Right, efficacy. Thank you. Thank you. Vaccine in the general population. I mean, the efficacy trials we do in clinical trials with the highly selected population. But once you start rolling out the vaccine and it's used in huge numbers of people, then you have to still monitor the vaccine and make sure that it is working in the real world among real people. And then you call that looking for vaccine effectiveness. So we should be quite amazed that within a very short period of time, after the virus was discovered or sequenced, that there were so many excellent vaccines, and some of them, I mean, all of them really, the mRNA vaccines and the adenovirus vector vaccines are actually new. They are first in the world type of vaccines, but they were highly efficacious. I mean, even these numbers are amazing. These numbers for highly efficacious vaccines that came out. So with those trials, we realized that the mRNA vaccines have very high 90 plus efficacies, and the ones with the incompetent vectors lower, but still highly efficacious. When you think of vaccines in general, these are highly efficacious vaccines. Then how about the other ones that came up after that? So we have the Sputnik, because the students were interested in what are the vaccines in Sri Lanka that are available. So we had the Sputnik that I said uses two different viruses. The initial adenovirus, initial vector is adenovirus 26, followed by adenovirus 5 as a booster dose. So according to the interim analysis, this one had a very high efficacy of, again, almost as good as the mRNA vaccines in preventing symptomatic COVID-19. Now, Sinopharm we are all interested in, because actually the majority of Sri Lankan population are getting Sinopharm. So it is an inactivated whole virus vaccine based on a SARS-CoV-2 virus isolated from a patient in China, with, of course, the adjuvants. And vaccine efficacy was still pretty high. It was pretty high. I mean, as good as the AstraZeneca, and it was estimated at 78% on efficacy trials. Right? And how about the Sinovac, very controversial Sinovac that was just approved just yesterday or day before, with 3 of the members of the expert group, in fact, resigning after it was the board overruled the experts and decided to approve the Sinovac. So again, the efficacy trials are still ongoing, really. Some of them have been published, and the reports are very, very variable, from 50 to 91%. So that is the problem. It's still not entirely sure how efficacious this vaccine is, because there's lots of conflicting trials, and there is still, still the data is not absolutely clear about the efficacy of this vaccine. Right. So now just moving on to controversies with vaccinations, the COVID vaccine. How does it differ from other vaccines, and what are the frequently asked questions on vaccination? So some people ask, now all of you have your hepatitis B vaccine, and after that, I hope after your 3 doses, you had antibody testing and you checked your quantified antibodies to make sure that you had, achieved seroconversion. So people might ask, shouldn't we document seroconversion? All kinds of vaccines are being given. Some are 90% efficacious, some are 50% efficacious. Should we not check that for seroconversion after vaccination? the problem is that the correlates of protection, like what actually protects you, what is the best marker of protection, are still not known. So we still don't know what the marker of protection is for us to really test and say, unlike hepatitis B, where we know anti-HBs antibody is the correlate of protection. Here we don't have a correlate of protection for COVID-19 vaccines. And the other thing is the serological tests that we use which are used, occasionally to diagnose past infection, are not probably the ones that will help us to identify an antibody response to a vaccine. Because remember, the vaccine is very targeted. It's only giving certain antigens of the virus, whereas after infection, you will have antibodies to a host of viral antigens. After a vaccine, you will only have antibodies to those particular targeted antigens that are in the vaccine. So the serological tests that we are using are unable to tell you whether you have responded to the vaccine or not. Then how about the next important question? Do these vaccines have any immunity against this variants? As variants of interest and variants of concern, and of course the alpha variant, which is the one that is really causing the third pandemic, and now we're having the Delta virus, which may in fact lead us from third into fourth pandemic pretty soon. So as we know, these vaccines were made for the original wild virus, and immunogenicity was proved against the wild virus. But now the virus has changed. And lots of the variants of concern are the variation is in the spike protein. So can our B and T cells actually combat these variants? So actually the data is quite limited, right? But in general, the WHO and most of the experts say in general that say alpha, which is the sort of common variant everywhere now, that in general COVID-19 vaccines retain their efficacy against these vaccines. So this is the what is known for the first lot of vaccines, and they seem to be quite good for the alpha variant and seem to protect against alpha variant. But you can see against the delta variant, there is not enough data. Now there's a little more data, and it seems that Pfizer against Delta was, just dropped from 93% efficacy to 90%. And for AstraZeneca, it seems that it has dropped from 66% against alpha virus to about 60% for Delta virus. So all the vaccines are showing us a drop in efficacy against the Delta virus, but it's still efficacious. I mean, even though there is a drop in efficacy, the efficacy is still there, but definitely it has moved down, a few notches of efficacy. So it's the same, the Sinopharm, Sinovac probably will also show this drop in efficacy. And you're starting from a much lower base. So you're going to drop, into quite a low territory with Delta virus. So that is a real concern. There's no doubt there's a concern. Then how about, do these vaccines have any impact on transmission? Remember, these clinical trials and even the effectiveness trials are mainly looking at hospitalization and fatalities, and really not looking at and also symptomatic COVID-19 with hospitalization and deaths. But how about asymptomatic infection, which is contributing to transmission? We still don't have the data on that. But accumulated data suggests that vaccinations does reduce even asymptomatic infection and therefore decrease transmission as well. Right. Then how about breakthrough infections? Everyone says, you get all these WhatsApps coming around, oh, this person had 2 doses of vaccine and they still got infected. Well, of course, none of the COVID vaccines are 100% effective. So you are expecting fully vaccinated individuals to develop infections. But anyway, basically these seem to be uncommon, and many of them are asymptomatic, right? So say in the mRNA vaccines, that is what is given in the United States, after 101 million fully vaccinated individuals, they had about 10,000 breakthrough infections, of which only 10% were hospitalized and 2% died. So in spite of, even though these vaccines are not 100% protective, they are quite, they do help you to, I mean, the breakthrough infections seem to be, even the breakthrough infections seem to be fairly mild. But certainly, remember, you can still die after 2 doses of vaccine. As you can see, 2% did die in spite of 2 doses of mRNA vaccines. Then, the other vaccines we are not sure, but of course, I mean, as you can see, the efficacies are much lower, say AstraZeneca at 65 or something. There'll be much more breakthrough infections, but we do hope that the hospitalization rates and the mortality rates will be low in these people who have both doses. The other question, of course, is duration of protection. How long will we be protected with our 2 doses? And really, nobody knows that. We know that there's waning immunity in natural infection, so there should, so there may be waning immunity even in vaccination. We know that the variants are also becoming more and more, escaping our immune responses. So the immune response is waning on one side, and the virus is becoming, more able to escape our immune response on the other side. So certainly now booster doses and booster vaccinations are being considered, and there are some countries that are going on to give a 3rd dose of booster vaccine, either the same vaccine or sometimes a different vaccine, to boost immunity in those who have been given just the 2 doses, the original primary 2 doses. Then vaccine safety. So far it seems that vaccine safety is pretty good for a vaccine that, was introduced so rapidly to the population. So most of them is just reactogenicity with little skin reactions and fever. But there have been serious adverse events. We know about the thrombocytopenic thrombosis that had happened with deaths. And they're also worried about, but there's something we don't know about. We don't know any long lasting adverse events. We don't know the long-term safety profile, and we don't know about, we don't know really how many people have had serious adverse events that have a lifelong impact. Then do you have to give a vaccine if you've been infected? Right, so we do know that natural immunity with coronaviruses is anyway short-lived, and although people who have been infected, it's uncommon for them to get reinfected, but we don't know how long they're going to be protected after natural infection, because with other coronaviruses, the natural infection, the immunity is quite short-lived. So the idea is that vaccination, when you give vaccination to someone who has been previously infected, it does boost their antibody levels massively. And so that the tendency now is to approve vaccination for those who have had infection. They should still be vaccinated. Once they've recovered from acute infection and have left quarantine and everything, then they are able to receive the vaccine. But remember, they might get more adverse events after the vaccine dose, because of course their immune system is already primed. Now, remember, there's other, you may have to get other vaccines. Some students who are, they may be still needing other vaccines, or pregnant women need their tetanus toxoid. People going overseas may need meningococcal vaccine. So how can you give this vaccine along with other vaccines? So though there's not a lot of data, generally the recommendation is that you can give the COVID-19 vaccine at any time in relation to other vaccines that you need. How about vaccines in pregnant women? I said that the original trials did not include pregnant women. Now the trials are having pregnant women, plus of course inadvertently vaccines have been given to pregnant women even in the original rollout. So what they have found is it seems to be extremely safe. And of course we know that pregnant women have a higher risk of severe illness and death. And in fact, some of the data from Sri Lanka shows that most of the children who have died of COVID-19, very few, but a majority of them have been less than 1 year. In other words, neonates and infants. So giving vaccine to the mother will protect the mother as well as the neonate, and therefore, we in fact we are targeting pregnant women as a group that should be vaccinated. How about the immunosuppressed? Well, again, immunosuppressed people are the ones who are at high risk of COVID-19 complications. And so therefore, again, we would like immunosuppressed people to get the vaccine.
Madam, excuse me, you are muted.
Sorry, when did I think it's just going on mute. From where did it go mute again, please?
This slide, this slide.
Okay, right. So vaccines in the immunosuppressed, yes, of course, because the immunosuppressed are the people who are most likely to get complications from COVID-19. We recommend immunization of immunosuppressed people, but of course the immunogenicity and the vaccine efficacy is uncertain in those people, and they must definitely practice all the other protection for themselves, because like in any other vaccine, we are not sure of the protection in the immunosuppressed in COVID-19 too. Right. So there are many unanswered questions that I am afraid I could not answer today, because we still don't know. And as things go on, day by day, we are learning more things about COVID-19 vaccines. So I'm sure in another month or two, we will know even more. And the best thing is to keep reading and looking out for the news. And you will learn, every day there's something new about COVID vaccines.
Thank you, madam. And also there are several questions asked from our viewers. One question is asking, does all these COVID-19 vaccines need 2 doses? And also another question is, 2-dose vaccine is enough for me?
Okay, so there's one vaccine, the Johnson & Johnson vaccine, where they have said it's a single dose vaccine. That is the only vaccine so far, I think, that is going as a single dose. The Sputnik people were trying to say, Sputnik also a single dose is enough, and the first Sputnik is enough. That's right. Someone has asked what is mini Sputnik. That's right. So they did produce this something called mini Sputnik, where they said a single dose is adequate for the Sputnik vaccine. But it's the same. The mini Sputnik is the same as the first dose of Sputnik anyway. So it is not something new or a boosted or anything like that. So we have to wait and see whether this mini Sputnik or a single dose of Sputnik is adequate. I think they took it from Johnson, where Johnson had a single dose, and were happy with that. Then somebody asked about 2 doses. I think I mentioned that at the moment 2 doses seems to be okay, but we are sort of watching this page. And in certain countries, certain countries have started to think about a booster dose, a 3rd dose. And that might really happen, because they've vaccinated everybody and they've given them their first 2 doses. And now they're kind of contemplating giving 3rd dose. And some people are giving 3rd doses. But I think the most important thing is that everybody should get their first 2 doses. So everybody in the world should first get first 2 doses before we start thinking of 3rd dose.
Okay, thank you. Thank you so much, madam. I think there are so many valuable knowledge that we have taken from that. And also, if you have any further questions, you can leave that from the chat. We can get at the end of the discussion. So, so then we are going to our next topic of today. I will get approached like this. One of my relatives asked from via call, what are the medicines you can use for a mild COVID disease who is under quarantine now at home? When she asked that question, actually I didn't have any knowledge about any medicine that we are using currently. So only thing I knew is there are no medicines for the COVID-19 until now. They actually doesn't have any medicine. So for that topic, I would like to invite our next speaker, Professor Sri Ranganathan. Madam, is there actually medicines we can use for COVID-19?
Thank you very much. Can you hear me?
Yes, madam, we can hear you.
Can you see the slides?
Yes, madam.
Thank you. So when I finish this lecture, if you still have the question, then I can answer the question through the chat. So thank you very much for inviting me to this webinar. I'm quite happy to share whatever knowledge I have on medicines for COVID-19 in this forum. So as you can see that you have arranged your lectures in a nice order. So we have spoken about the vaccines by Professor Inoka Korea, and you are going to having a very entertaining lecture about how to manage a patient with COVID-19 in Sri Lankan context after myself. And I will be speaking only about current status of medicine for COVID-19. Unfortunately, that we have two pandemics ongoing these days. So number one is the COVID-19 pandemic. Other one is the pandemic of misinformation and fake news. As you can see in the bottom, that when I searched for COVID-19 clinical trials, I got 287 million hits in about 79.79 seconds when I did this about 4 days before. So therefore, I have relied only 3 information sources in my presentation. So number one is the World Health Organization. As you can see, there is a COVID-19 therapeutics guidelines, living guidelines is being published continuously by the WHO. So I will be using that for this presentation. And based on that, the British Medical Journal has been regularly publishing a rapid recommendation, like what you like, because it is a graphic with so many colors, not like our lectures with a lot of bullet points. And also because I wanted to make it little simple, because some first year students or second year students might be joining this webinar, I also used a medical journalist view of coronavirus treatment, which appeared in the New York Times. So these are the 3 reference resources I have used in this lecture. As you know, rational management of any diseases have 4 strategies. One is you have to provide information to the patient. Second one, you have to treat several methods of treatment without medicine, that is called non-pharmacological. And the third one is treatment with medicines. And the fourth one is if you cannot manage with all these strategies, you can refer the patient to a higher level. So the objective of my lecture is to discuss with you what are the options that we have to treat a patient with COVID-19 for pharmacological treatment. I classified them into 4. One is medicine against the causative agent. Second one, medicine acting on the pathophysiology of the disease. Third one is antibacterial for prevention and treatment of bacterial complication if there is one. And the fourth one is medicine which reverse or control non-infectious complication. And in this presentation, I will be focusing only on the first 2. And interestingly, the New York Times journalist also has classified, as I have classified almost. One is the blocking the virus, so that is the acting against the causative agent. Second one, he or she is writing about mimicking the immune system and putting out the friendly fire. I will come to that later. So they are the medicines which will be acting on the pathophysiology of the disease. And the last one is the other treatment. But I want you to have your attention on the figure on the left side. As you can see, up to July 8th, these journalists have been tracking the medicines which are being tested in COVID-19 from the onset of this pandemic until July 8th. There have been only 1 medicine which has received the FDA approval, that is the Remdesivir. I will come to that. And you can see 13 of them actually have mixed evidence, or how do you call it, tentative evidence. So as you can see from here is that we don't have evidence, which now in the era of evidence-based medicine, to have effective medicines for COVID-19 other than about 1 or 2. And as you can see, why they call that it is tentative or mixed evidence, because the research evidence comes from animal studies or from retrospective studies, or even in a prospective clinical trials, the results are differing from one study to other study. Because if a medicine is truly effective, there should not be inconsistencies between different studies. So all of them have been lumped into mixed or tentative evidence. Whereas the one which has FDA approval or promising evidence is at least in one randomized controlled clinical trial, that particular medicine fared better than the placebo. So this is the summary of the medicines that I would be speaking today. And this is what I explained to you, that is what is meant by promising evidence and what is meant by tentative evidence. So now we will look at the first group, that is the medicines acting against the causative agents. Unfortunately, the super scientific humans was not able to discover any effective antiviral agent for the viruses which have been causing the pandemics in the recent past. As you can see, the figure that I have put it on the left side. So most of the viruses which hit the world in the recent past, not recent past, in this decade probably, that we have not discovered an effective antiviral agent. Let's look at what happens to the COVID-19. This, I have listed about 16 agents which have been investigated for possible antiviral activity against this virus. Only one, that is the Remdesivir, had some form of promising results. And the purple one are called as tentative or mixed evidence. And the red ones are not promising. So up to now, in this 2-year battle between the super scientific humans and the tiny coronavirus, the virus still is the winner. So let us look at about this Remdesivir. So this is the first and the only agent that gained approval from the FDA for the treatment of coronavirus. It was originally tested as an antiviral agent for Ebola and hepatitis, but with disappointing results. They were not working. So when the pandemic started, the industry started to test this medicine against COVID-19 virus. And the proposed mode of action was it will act at the viral gene level and it might prevent the multiplication and replication of the viruses further. So the first clinical trial reported the Remdesivir reduced the duration of hospitalization with COVID-19 from 15 to 11 days. So that is the first data which came up for the efficacy of Remdesivir. This is not on mortality or morbidity, but it is on the duration of hospitalization. But based on these results, because there were no alternative medicine, the FDA gave a emergency use authorization. I will tell about that at the end of my lecture for critically ill patients who are hospitalized with supplemental oxygen. Later on, as you know, the pharmaceutical industry, they are not going to stop with using this medicine only in critically ill patients. So they extended their clinical trial to less severe form and also extend their treatment to 5 days so that the profit from the sale of the product also is going to be skyrocketing. And the conclusion was, as you can see what I have underlined, seem to benefit modestly the less severe forms of coronavirus. So you can mark the word seem to benefit modestly. However, based on this event in August 2020, FDA again extended its approval for any severity of the diseases. In October 2020, the then president of the United States was affected by the COVID-19 infection, and he received a lot of medicine. Remdesivir was one of them. And with that, actually, FDA actually gave approval for this use in pediatric about 12 years, as well as in adult. However, as you can see, in United States, a 5 days course cost about $3,000 USD, and the company earned $2.8 billion US in 1 year, and the company boasted 1 in 2 Americans hospitalized for COVID-19 was receiving Remdesivir. However, soon after that, in November 2020, the WHO actually collated all the clinical trials which has been done on Remdesivir and found that it is weak recommendation against the use of Remdesivir in hospitalized patients, regardless of disease severity, as there is currently no evidence that Remdesivir improve any outcome in these patients. You should know registration or approval of a medicine by a regulatory authority is actually not equivalent to recommendation for use in a World Health Organization guidelines. The criteria used for approving the registration and adding a medicine in a recommendation are really different. So why the WHO didn't make that recommendation? So this is the Remdesivir. I will be putting this type of slides in my subsequent slides as well. So try to work out where you can see. So that blank one is the one that is the, the blank one is the one that you should focus. So here weak. And also it is against the Remdesivir, as you can see. So it has to be given intravenously and initially on day 1, and their subsequent course was up to day 5. Initially 200 milligram and later on 100 milligram, but the WHO recommendation was to suggest not to use Remdesivir. Why is so? Again, can you look at on the top left side on important outcomes, mortality, mechanical ventilation, serious adverse events, viral clearance. As you can see, time to clinical improvement, all are important outcome of treating a disease. There was no difference between the Remdesivir as well as the placebo, that is a standard supportive care. In addition to that, WHO noted it is an intravenous administration. optimal timing and dosing was not known. And also it's quite expensive if the branded product has to be used in lower middle income countries as well. So based on all those things, WHO actually did not recommend use of Remdesivir. However, under values and preferences, it has suggested individual clinicians and patients, because this is a global disease, patients are also well aware about these illnesses, that they can choose to use Remdesivir even if they have not recommended, because the evidence has not excluded the possibility of benefit, because it is no difference, right? It has not favored the Remdesivir, it has not favored the placebo also. So with that Remdesivir, that there are other antiviral medicines which have been tried in the treatment of COVID-19. The second and the fourth one, there's a tongue twister for you to pronunciate them. So it's all, both of them finishes like Piravir. So they also have not shown any efficacy on this virus, negligible impact on mortality, as well as the next one, the last one. Actually, the sponsor has to stop the trial because the data showed that it is unlikely to help. But the third one, you have to understand, this virus goes and sometimes binds to the angiotensin converting enzyme receptor in the lungs. So the industry were trying to develop a receptor blocker for this particular receptor so that the virus will not attach to it. Up to now that we have no data on humans about this particular product. Then, as you know, you may have seen, because social media is publishing more than the traditional journals. So ivermectin is actually an antiparasitic. It is not antiviral, because there is no human data at that point that to call that it is an antiviral. In April 2020, Australian group of researchers published data. It could block the virus replication in cell cultures, but they used a fairly higher doses. In in rich countries, ivermectin is not used on humans. We are in Sri Lanka, we have used it for filariasis, but in rich countries, they were using for parasitic infections in their pets. So in April 2020, FDA said not to use this pet medicine for COVID-19. And in March 2021, again, they stressed not to use it for treatment or prevention of COVID-19. EMEA, that is the European Medicine Evaluation Agency, also published a similar warning. NIH is the guideline published by America. It also said it's insufficient data. In fact, USA, instead, there was an inquiry by the Senate, because one doctor has been claiming that it is a miracle medicine in the treatment of COVID-19. So as a global pandemic hits all of us, the countries do not differ in their claims. As you know, in Sri Lanka also, we had so much of claims about miracle medicines, but in the USA, there had been a Senate inquiry and the doctor is being sort of disciplinary action has been taken. However, with all these things, the World Health Organization looked into the availability of data for ivermectin and found that there is no data to support its use in COVID-19 patients up to now. However, this can be used in clinical trial settings in order to provide the data. Because of that, the clinical trials in ivermectin is still continuing to see whether it is going to favor or whether it is going to be against the use of COVID-19 treatment, as well as because there is no standard dose for this. So because this medicine was not developed for COVID-19, this was developed for some other parasitic diseases. So up to now, no one knows what is the correct dose for this medicine. So therefore, clinical trials are still continuing. Why WHO discouraged use the ivermectin? As you can see, that it's favors the supportive care stronger than the ivermectin. And if you can see on the right-hand side bottom, important outcome, there is no important differences, but the third one, that is a viral clearance, is faster with the ivermectin, but that was not one of the most important clinical outcome. But as you can see, serious adverse effects were more with ivermectin, 18 more with ivermectin than the placebo. But what actually made the WHO not to recommend this also, there were only about 900 patients who have been subjected to clinical trial for this medicine, and there have been only 31 deaths. So they thought that to make a conclusion based on 31 deaths on either side, either placebo side or as well as you can see about 70, and the other side is about, I think, I can't see that mortality. So it was about 31 deaths. So therefore that they were unable to make a conclusion. So that is the reason that the WHO has not yet approved or recommended the use of ivermectin as an antiviral agent. So why this problem? I think Professor Inoka Korea would know better than me. Viruses are different from bacteria. They don't have a cell wall. They don't have a metabolic pathway. They go inside the human cells and depend on human proteins to work for them. And there are very few natural antivirals which we can modify and produce as an antiviral agents. We have to preserve the humans when we give antiviral medicine, because these viruses are inside the human cells. Only thing that we can do is we can burn both. That is, we can burn the human cells together with the virus inside, which you can't do. And also the emergence of mutants. So now you heard from Inoka what the problem with related to vaccine, and for medicines also emergence of mutants. So this summarizes the problem that we have in discovering antiviral medicines for coronavirus. Viruses basically act like a thief inside the cell. They steal the cellular machinery of humans and repurpose those machines to make their own copies. They don't have any of the hallmarks of living things. So therefore, we don't have a target in the virus for us to develop medicine against the virus. So let us now move on to the second group of the medicine. So as you can see, acting against the causative agent, only one is approved by the FDA, none is recommended by the WHO. Now we are coming to the medicine which against the pathophysiology of the disease. So almost all of them who are joined here must be the students. Then you know, say for example, even meningitis, there is a role for using steroids in the treatment of meningitis. Like that, they have a clinical trial going on to see whether they could discover or recommend medicine which could act on the pathophysiology of the disease. So as you can see, the New York Times journalists have divided that into mimic the immune system and putting out friendly fire, because the friendly fire is supposed to be the inflammatory process, but then it becomes a disastrous consequences also. So the medicines which are mimicking the immune system, there's a long list which shows the human panic against the tiny virus, showing us there is a lot more than the pharmaceutical world that we have. And there were three in that category that I can speak of, which you have heard about also. Number one is convalescent plasma, number two is the monoclonal antibodies, and number three is the interferon. However, interferon, still the researches are going on. There is no clinical trial as such, and there is no emergency use authorization by the FDA also. So let me tell something about convalescent plasma and the monoclonal antibodies. Convalescent plasma is a form of passive immunization, you know, anti-rabies serum, anti-tetanus serum, and in the past anti-diphtheria serum, where you try to give the antibodies isolated from the people who have recovered from this infection. There is a long story in the United States, like all the other medicine, because they were badly hit by disease which they were not familiar of and which they couldn't have an alternative treatment. They were actually taken by surprise. So there are long stories of these medicines in the United States. It has been used without clinical trials. However, when the researchers started to do the clinical trial on convalescent plasma, they have to stop the clinical trial, both in America as well as in UK, because there was no benefit. So they didn't want to continue to recruit patients on this clinical trial. But there is a small trial coming from Argentina. It is a small trial rigorously conducted. This could prevent some early, if you present early, this could prevent the patient going into severe diseases. So because of that, FDA actually revised its emergency use authorization for plasma. Only for plasma with a high concentration of antibodies can be used. And also that is recommended for hospitalized patients who are unable to develop the antibodies on their own, that is the immune suppressed group. Then about monoclonal antibodies, you must have heard, at least the senior students who are in final year and the fourth year, you would have heard use of monoclonal antibodies in cancers, monoclonal antibodies in arthritis, because as opposed to plasma, which is polyclonal antibody and also developed in the humans, it is actually developed in lab. And also this is monoclonal because it targets only one section of the immune process. However, this monoclonal antibodies is considered as a discovery of this decade. Since 1970, FDA has approved monoclonal antibodies for about 79 diseases. Actually, it is developed in a laboratory where one, maybe few collection of B lymphocyte is exposed to a particular viral protein. In this case, it is the spike protein, and then it is cloned to mass produce antibodies in the laboratory environment. So this is how the monoclonal antibodies for various invaders, cancer cells or infectious agents are being produced. So for the coronavirus, actually, it is the spike protein against which the monoclonal antibodies are being developed. And as you can see in this figure, this is how it works. As you can see, the spike protein in the virus, and then there are antibodies for this spike protein. So when they go and bind to that spike protein, then the virus cannot bind to the lung cells or the other human cells. However, you all can work out this to happen, we had to give this medicine quite early before the virus can bind to the human cell. That itself will be a problem. So that will be a one negative factor for these antibodies, because it is costly and availability of this in many hospitals would be limited. So giving early would be difficult. Again, it has to be given by infusion. So it takes time to reach the site of action. Again, that is a limitation. But the major two limitation about this monoclonal antibody is it will not be effective for new variants, because we have developed that for a previous variant for spike protein of that particular variant. And if the new variant has a different structure for the spike protein, this medicine will not work. And the other disadvantage is the cost. So now you can see that three monoclonal antibodies. So the first one is actually two antibodies manufactured by, I think, Eli pharmaceutical industry. So one is the actually it's a tongue twister, so bamlanivimab, and other one is the etesivimab. FDA gave approval for the first one initially. When a lot of emergence of variants came, then they gave approval for both of them together, insisting that both together have to be used. But as you can see, recently in June 2021, they revoked those approval because of the rise of the beta and gamma variants. The next one, which has actually received a lot of fame and popularity in India, and also have received the emergency use approval in Sri Lanka, has a cocktail. So they are together two types of monoclonal antibodies. Each claims that it is effective for the beta variant as well, but it has not been shown. However, it has an approval in Sri Lanka as well, because we thought that it could work. I thought the regulatory authority thought it could work for some of the variants which are existing in Sri Lanka. So the monoclonal antibodies also, the WHO is still working on the monoclonal antibodies. They are not released their guidelines yet. But what I want to say is that when I was reading for this lecture about a week before, this particular paper was not there. Again, when I started to read for this particular lecture, day before yesterday, this paper has come. So this is the first monoclonal antibody, because the pharmaceutical industry, who have invested so much of money on these products, is not going to just stop just because a new variant has come. So they work hard to publish some form of efficacy data so that they can encourage use of these medicines and they can get their investment back with a profit. So this paper published on 14 July 2021, and which claims it might be useful in mild to moderate COVID-19, but I couldn't read the entire paper, so I didn't see what they argue for the new variants. So when you go to the second group, medicine which put out the friendly fire. So as you know, the inflammation, which is supposed to be beneficial for the patients, sometimes produce some harmful effect as well. So this group of medicines were actually thought might working as anti-inflammatory and would reduce the progression of the disease. So this is why these three groups are there. One is the dexamethasone, other one is cytokine inhibitors, other one is the interleukin-6 inhibitors. So WHO actually has recommended the dexamethasone as well as interleukin-6 inhibitors. Those are strong recommendations. The first recommendation came somewhere in September 2020, and the interleukin-6 inhibitor, that is the tocilizumab and the sarilumab, came just on July 6th, as you can see. But these two medicines, we don't need to worry about the regulatory approval because they have been already approved by the regulatory authorities for some other indication. But in Sri Lanka, tocilizumab was approved for probably the arthritis, but sarilumab not yet. So therefore, we have the dexamethasone and we have the tocilizumab. So because I'm going to speak about them little later, I have just listed the other medicines which have been actually investigated, thinking that they could play an anti-inflammatory role. As you can remember, azithromycin was heavily promoted in the initial pandemic period, but then a large-scale randomized trial said that there is no benefit. Same for colchicine, which was investigated in a small clinical trial, but later on they saw there is no difference in mortality outcome. Then the other, leronlimab, XOCD24, stem cells, and if you can see number 8, those who have gone through mental health module know fluvoxamine is a selective serotonin reuptake inhibitor used in the treatment of depression. You can see how panicking the humans are. So they have even tried SSR, I think, might have some anti-inflammatory effect. So a small RCT has claimed that it reduced hospitalization rate between 8.5 to 0. So a large RCT is ongoing. So now let me tell you why dexamethasone and the tocilizumab were recommended by WHO. These documents are openly available. I send them everything to the group which is organizing this workshop. So as you can see, these are out of the 6 medicines the WHO worked on. I'm fortunate to be part of the team that is working on these guidelines. So as you can see, out of the 6 interventions, only 2 had been recommended by the World Health Organization. Both of them act on the, how do you call this, friendly fire, that is the extensive inflammation. And now why the corticosteroid has 2 recommendations. The recommendation 1, it is beneficial for severe illness and critical illness. It is not beneficial for mild illness. So you can see these slides later. Corticosteroids can be dexamethasone, hydrocortisone, methylprednisolone, or prednisolone, and the duration is about 7 to 10 days time. And the evidence which supported its recommendation, as you can see on the left-hand side upper one. So in a critical one, the death was 87 per 1,000 fewer. And in the severe one, 67 per 1,000 fewer. Only problem that they noticed is there is about 46 per 1,000 higher rate of hyperglycemia when we are using corticosteroids. However, in 2021 March, the British government claimed that the dexamethasone has saved 1 million of lives in their country. However, the WHO also noticed that these medicines are freely available in all the countries, as well as that the adverse effect associated with steroids are generally happen with long-term use. So they recommended this medicine in somewhere September 2020. Now, why didn't they recommend it to use in mild diseases? As you can see, that the mortality rate is actually little higher when we give the steroids when the disease is mild, because most probably by suppressing our immunity, the evidence what we have is actually the mortality is a bit higher when you use corticosteroids. So that is what it is very important to categorize patients whether they have gone into severe or critical state. Then why did they recommend this IL-6 receptor blockers, that is interleukin-6 receptor blocker? 2 medicines have been researched. One is the tocilizumab, another one is sarilumab. And as you can see in the bottom one, that they have been 15 per 1,000 fewer deaths and 23 per 1,000 fewer mechanical ventilation. Duration of ventilation was 1.2 days fewer, and duration of hospitalization is 4.5 days fewer. So because of that, as you can see, the white one is the recommendation. They strongly recommend the use of IL-6 blockers. They are given intravenously. 1 dose is given over 1 hour, but the clinician can decide to give a second dose if they think that it is required. So, but the problem they want was 2. Number 1 is it will be expensive, but fortunately Sri Lanka, we have tocilizumab, but I know now it is not available for patients with arthritis because it is being with COVID-19. And also there is a rare possibility of causing secondary infection. So how these 2 medicines act in, I will probably take about 5 minutes more. How these medicines act. Dexamethasone, I don't need to tell you, all of you would have learned when you learned about meningitis. So because steroids, it's as you can see here, there's when there is this, So in the disease, what happens that they increase the pro-inflammatory cytokines and reduce the anti-inflammatory cytokines, which is responsible for the cytokine storm. So dexamethasone or all the steroids, they have the actually cytoplasmic receptors. So they go inside, bind to the cytoplasmic receptors that A-level 2019 also know because I taught them, and our foundation module students will also know 2018. So as you can see, that goes inside, then actually at the level of the genetic transactivation. So it reverses what the virus does. It increases the anti-inflammatory cytokines and reduces the pro-inflammatory cytokines. Then how does this tocilizumab act is, as you know, again, this you can go through that, but this interleukin-6 is binding to the receptor. It has a soluble receptor. So binding its soluble receptor, and together they come and bind to the G protein 130 actually initiate the tissue injury. So what the tocilizumab does is it actually blocks the soluble receptor so that interleukin-6 cannot bind to the receptor, and those 2 complex will not be binding to this GP 130 and will not be initiating the tissue injury. What are the safety concern related to this medicine. Dexamethasone, other than hyperglycemia, nothing is there because it is short duration. IL-6 receptor blockers, we don't have evidence about secondary infection because all the clinical trials have been done in developed country where the rate of baseline infection is quite low, like TB or silent TB remaining there. And also that they have excluded almost all the patient with active infection than the COVID-19 during the clinical trial. So the data from lower middle income countries have to be provided to see whether it has any secondary infection as an adverse effect. Then based on the WHO guidelines, I just thought the students would like to know, WHO has made a conditional recommendation for use of home pulse oximetry to pick up the patients who are progressing into severe or critical stage as early as possible. They also have recommended awake prone position, and also they have recommended the use of anticoagulants, but in the dose of thromboprophylactic use, not in the therapeutic or intermediate use. And as you know, again, the social media you would have seen, and also in the clinical setup, that is what I wanted to make a note here, that what I am presenting is the evidence-based medicine which is available in the literature, but the clinicians who are treating patients in a very difficult situation have more, when there are no option, they will opt to select whatever the treatment they believed and they have experience thinking that it will be beneficial to the patients. So you should not confusing the evidence that I am presenting and the clinical scenario that you would be witnessing. But if you work according to the published guidelines, then you would be using only the medicine which are included in the guidelines. So vitamin D, vitamin C, and zinc, as far as possible, there is no evidence from randomized controlled clinical trial showing the product is more efficacious than the usual supportive care. And vitamin D, why it came up is there is an association between low level of vitamin D and rates of COVID-19. But just because the people with low level of vitamin D have high rates of COVID-19 does not mean giving large doses of vitamin D is going to actually prevent patients getting COVID-19, because this is that association and not causation. So with that, I want to show what is happening in Sri Lanka, the medicines which are available. All the steroids are available. Tocilizumab is available. Sarilumab is not yet available. I heard there is some problem in the registration. Remdesivir is not registered. In the monoclonal antibodies, as you can see, one of them is registered in Sri Lanka. I repeatedly use two terms. One is called emergency use authorization. That happened in United States. What you mean by emergency use authorization is not a formal approval by the regulatory authority. This is if the government declares the particular condition is emergency, then the FDA use a different yard scale and give emergency use authorization, which can be withdrawn when the situation changes, not like a formal FDA approval. In Sri Lanka also, I saw that there is a guidance on emergency use permission, which is for vaccines, but I'm told that is used for medicines as well. It is available in the web. Then I used another term called conditional or weak recommendation by the WHO. WHO now uses only 4 recommendations based on strength, whether it is weak or strong, and based on direction, whether it is for or against. So there will be strong against, weak for, and weak against. So that would be the 4 type of recommendation the WHO would be making. So they have so far made 2 strong recommendations for, that is a corticosteroids as well as the IL-6 receptor blocker. And they have made strong against for about 4 of them, which you have seen there. And they have 1 week against, that is the use of corticosteroids in mild COVID-19. And they make this decision based on the balance between desirable and undesirable effects, based on the quality of evidence. And in addition, the preferences, values, and the resource consideration, like whether it is an intravenous preparation or an oral preparation, also taken into consideration by the World Health Organization. So what you mean by weak is if less confidence of the balance between desirable and undesirable consequences, a weak recommendation is made. However, that is confused with weak evidence. The people like the term conditional rather than the weak recommendation. So upon the patient values and the clinician's preferences, even if the WHO makes a weak recommendation, the clinician or the patient can still request this medicine to be used. That is the weak. So the last but not the least, the question which was put forward by the person who was, by the student who was moderating this, caring for someone sick at home. There are only 4 steps here. Isolate the sick patient as much as possible within the home setting. reduce the contact with the virus as much as possible, take care of the sick person in terms of antipyretics, increased fluid, nutrition, monitor symptoms, and adequate rest. There is no other medicines. And call the healthcare provider immediately if you see any danger signs. So this is the flyer by the WHO. As you can see, there is no medicine recommended for mild COVID-19 disease based on the existing evidence available in the literature, other than giving antipyretic, fluid, and nutritious diet, and keeping the person isolated and reducing contact with the person. Thank you very much for giving this opportunity. I think I have taken a little bit more time, and thank you for patient listening. If I have questions, I will answer through the chat. Without wasting more time, please call the next speaker. I will answer the question during the chat. Thank you very much.
Okay, madam. Thank you for your valuable knowledge. I think we would get a vast kind of knowledge in and out of these medicines that we are using. So, so as madam said, you can leave questions from the chat, and she will answer them through the chat also. So let's go to our next speaker, Dr. Harsha Sadishchandra. Sir, since the COVID-19 patients are managed in the separate wards and separate hospitals, as medical students, we didn't have any kind of approach, or we didn't have seen any patient in the wards, and we don't know how these patients are managed, what happens in the ward. So can you give us a knowledge how these patients are managed in the wards and how these patients are treated from the health staff in the hospital? So this is your chance to give this knowledge to us.
Yeah, thank you, Naveen. I will first try to share my slides with you.
Okay. We can see slideshow.
Management balance interesting.
Are you all right? Can you see the slides?
Yes, sir, we can see.
Yeah. Thank you very much. I must thank Prof. Shalini and Prof. Inoka for setting the scene for my talk. I think you have set the background nicely, so it makes my task much easier trying to discuss the management of COVID-19. So I will place special emphasis on the management of COVID-19 in Sri Lanka, because that may be what you're interested in. And as Naveen said, I think he wanted to know how these patients are managed in different settings. So I would try to do that. And the management I have actually based on the Sri Lankan guidelines. Now, there is a guideline which we follow, which was issued by the Director General Health Services in late May in the form of a circular. So it's quite a binding document. It goes beyond the guideline. It's a circular, so which we have to follow. And that is based on various expert groups working on the evidence and trying to devise a set of steps that are evidence-based and also practical, and practically in our setting. So if anyone wants to sort of get more details about it, I think you could check on that in the ministry website. And it comes under the heading Revised Clinical Practice Guidelines for COVID in Sri Lanka. So that's a good reference that we could have. Now, categorization of COVID-19, and you may know that there are different presentations, and I think this has been alluded to even by the previous speakers. There are asymptomatic patients, then there are patients with mild to moderate disease, patients with severe disease, and patients with critical disease. So like in dengue, in dengue, we talk of different presentations. So even in COVID-19, we do see these different presentations, and it's important to understand the stage of the illness, because the management is based on the stage of the illness, plus of course, the clinical signs. Now, if I talk about the asymptomatic patients, now asymptomatic patients are actually in the early part of the pandemic with the what we call the wild type virus. The asymptomatics were quite a high percentage, and quite close to about 30 to 40% were asymptomatic patients. But now we do see increasingly more symptomatic patients. And with these new variants, as was discussed by Prof. Inoka, with alpha, beta, and delta variants, we see more symptomatic patients than before. So one has to be a little careful when we say that asymptomatic patients are common now. But again, another point is not all will remain asymptomatic, and some may be just pre-symptomatic at the stage we pick them up. And if we monitor them, they go on to become symptomatic at some stage. And the important point to note is that these asymptomatic patients are infectious, and they could secrete high viral loads. So they form an important part in driving the pandemic. So this is something which we haven't seen with other respiratory viruses. Of course, we do see that even with influenza, but in COVID-19, this is a major feature. And if you take the SARS coronavirus 1, the SARS epidemic, which happened, I think, in 2003, then we did not see asymptomatic patients, and the patients developed illness after about, after a fairly lengthy incubation period. And we could isolate these patients and control the pandemic. But here we find it difficult to control the pandemic because there is a sizable percentage of asymptomatic patients driving the pandemic. So, but these patients, as I said earlier, do need monitoring to detect progression of the disease. And as was told by Prof. Shalini, they do not need any specific treatment. They don't have symptoms. But we currently, the Sri Lankan policy is to isolate these patients at intermediate care centers. I mean, by intermediate care centers, what we mean is, they are sort of lower level hospitals where the patients are sort of taken care of. And there are medical officers and there are nurses, but they are not high category hospitals, and they are sort of limited in their facilities. Now, it is important to know that there's a pilot project going on in the Kalutara district to have these patients at home and monitor them at home. So, I mean, I think you would understand that most of the patients do not develop severe disease. So we say close to 80% of the patients do not develop severe disease. So they can be, if facilities are available, monitored at home. But one important thing is that some of these patients may develop symptoms and may develop complications, so they have to be monitored wherever they are, whether it's in intermediate care, whether it is in an intermediate care center, or whether it is at home. So there has to be a monitoring mechanism, and the project at Kalutara, I think that has gone on for about a month now. And I was recently fortunate enough to listen to a presentation about the outcome, and it seems to have worked pretty well. And so it may be extended to other areas in the Western province in future. Now, mild to moderate disease, the common symptoms of COVID-19 are fever, dry cough, shortness of breath, headaches, myalgia, and then this one peculiar symptom, loss of smell and taste. It's not there all the time, but if it is there, it is said to be quite characteristic because you don't really see that in other illnesses. Then there could be sore throat. There could be GI symptoms as well, like diarrhea. So there can be atypical presentation. Diarrhea is mostly towards the end of the first week. Now, we do know that there's a range given as the incubation period for COVID-19 from 2 to 14 days, it is said. But what we have seen now from data around the world is that the incubation period is actually is much shorter than that in most patients. Of course, there can be the odd patient who will incubate for 1 week or more, but mostly it's about 5 days. And they are infectious 2 days before the appearance of symptoms. And the rate of infectivity persists up to about day 5 after the appearance of symptoms. So it's in about a week that these patients are quite infectious, and that is the period when they spread the virus quite rapidly. Now, with these new, with the emergence of new variants like the Delta variant and of course the Alpha variant, there was talk that the symptoms were different, but actually the symptoms, I mean, most of the initial symptoms are still seen. So I don't think there's a big difference in symptoms. But what we do see is that more patients with the Alpha variant and the Delta variant do seem to develop severe disease, whether it is due to the higher infecting viral dose or whether it is due to an increase in virulence of the virus per se is not very clear. So there is no one is very clear about that. But we do see with the newer variants, higher percentage of patients developing symptoms, higher percentage of patients needing oxygen, and higher percentage of patients, unfortunately, perishing in the illness. So I think it's important to know a bit about these clinical factors. Now, the viral load peaks on about day 3, and then it declines. So after about the first week, if the patient clears the virus, then the patient is not infectious, but of course the patient develops pneumonia and complications, then the patient can remain infectious for longer. So there won't be any lung signs in mild disease. And again, even for mild to moderate disease, there is no specific treatment. Rest, paracetamol, good hydration, good nutrition, sleep. I mean, those are the main modalities of treatment. No specific treatment is there. Then in moderate disease, you may, in moderate to severe disease, you may hear lung signs, and usually they are in the form of crepitations in the lung bases, in the base of the lung. And the chest X-ray actually may show peripheral and basal shadowing. So that's a bit of an unusual thing, because sometimes in other illnesses, you know, the patient has to look quite ill for them to develop pneumonia based on X-ray findings. But here we do see some patients who have lung signs, and so when you listen, you detect crepitations, but they do have X-ray changes as well, but they look well. So that is a peculiar thing with this virus. And if HRCT is done, that will show what we call ground glass opacities in both lung fields, again in the basal and peripheral parts. But even with moderate disease, most patients will improve by day 7. Now, this day 7 is a critical point. Now, I think we do talk of a critical period in dengue. And similarly, we do see that patients who get the virus, who are infected with the virus and who display symptoms, by about day 5, they begin to recover. And by day 7, most patients recover and they feel better. But the percentage of patients who go on to develop severe disease and critical disease will deteriorate after day 7. So that is, day 7 is a critical point in the illness, and we have to monitor these patients very carefully at that point. And oxygen saturation must be monitored, and the patient should be transferred to a higher grade hospital if the saturation is low. Now, one thing about oxygen saturation, now these patients do appear to tolerate hypoxia well, and there is this term called happy hypoxemia, where the patient is lying comfortably at rest, and they may, and if you happen to have a pulse oximeter and check their oxygen saturation, that may be actually may be low. Now, normal person should have a saturation over 96, but these patients may have even 90, but they don't seem to show it. But if you get them to take a few steps, if you get them to exert a little bit, then they desaturate fast. So in our centers, we do a thing called a supervised exertional test, where the patient is asked to take a few steps, or the patient is asked to sit and stand on a chair, or rather sit and stand and sit on a chair, say about 10 times within a minute. And then we check the saturation to see if there's any drop. So some of these patients actually are quite okay at rest, but when they do exercise, their saturation drops. So that is a sign of severe disease, and those patients have to be transferred to a higher grade hospital. Now, this is what I told you about the X-ray finding in COVID-19. Now, this is, I don't know if you can appreciate that, the X-ray changes are mostly in the basal and lateral parts. The upper lobes of the lungs are quite clear actually, but towards the basal part and the lateral part, you see shadowing. So this is a characteristic X-ray of a COVID-19 patient. I think now people have actually, unlike in other illnesses, COVID-19 is especially in that way, it gives characteristic radiological findings, both in the chest X-ray and in the CT scan. So much so that not just the PCR, but these radiological investigations are also taken into account when we treat these patients. And it's useful in diagnosis as well. Now, this is a CT scan just for your interest. Again, the changes you see are mostly towards the peripheral part and in the basal part of the lungs. So that is, we don't do CT scans on each and every patient, but when we do the CT scan, we do see these changes. Now, there are patients who are at high risk of developing severe disease. Who are those? Those over the age of 60, pregnant women, and very importantly, those with comorbidities. Now, they could have ischemic heart disease and different other types of heart disease, diabetes, hypertension, chronic kidney disease, chronic liver disease, chronic lung disease, smokers in that context, severe obesity. So those are the conditions in which the disease runs a severe course. And it's important that the thing that we do see is that the patients who develop severe disease are those with these illnesses with poor control. Now, there are patients with diabetes who have good control and they do better. So it's a very important message to take to the community as well. People with these comorbidities, they should control their comorbidities better than they ever did to get to sort of be able to sustain a COVID-19 infection. So that is an important part I think in the Asian context. We see lots of patients in their 40s, 50s having these comorbidities. So that is probably why we do see more patients in that 40 to 50, 40s and 50s age groups as well, who go on to develop severe disease and complications. Then those who are immunocompromised, those on long-term immunosuppressive therapy, obviously. So these patients, actually, sometimes we do not manage in these intermediate care centers. We send them to level 2 hospitals, which are sort of larger base hospitals. And sometimes some of these patients are managed in dedicated COVID centers like the infectious disease hospital. So these patients have to be taken care of in a special way. Now, severe disease is when patients have significant COVID-19 pneumonia, and then they have tachypnea with a respiratory rate of over 30 per minute. So they have lung signs, bilateral and basal crepitations, and the chest X-ray will show the abnormalities that I showed. And when the saturation at rest drops to 94% or less, then we initiate specific therapy. Now, I think Prof. Shalini gave an excellent account on what drugs are beneficial in COVID-19, citing evidence. And we did hear from her that there is no single antiviral agent that has shown mortality benefit, which is the issue that we are facing now. But there are lots of other agents which can reduce mortality in COVID-19. So these agents have to be initiated at this critical point. Again, a critical point here, when the saturation of a patient drops to 94% or less on air at rest. So that's the point that we are looking for. The moment the patient reaches that point, the patient has severe disease and the patient will benefit from our specific modalities of treatment. And if we don't give those modalities of treatment, the patients can deteriorate. So there are actually 5 pillars of treatment of severe disease, severe COVID-19. So these are very important to categorize. Oxygenation and fluid management, very important, because these are things that we sometimes tend to sort of not give special emphasis to, but the general management of patient is very important, because we have actually treated viral pneumonia in the past. I mean, before COVID-19 came into existence, there were patients with viral pneumonia, although not to this extent, and they were taken care of with supportive treatment. So we have to highlight the importance of oxygenation and fluid management. Steroids, anticoagulation, immunosuppression, and antimicrobial treatment. So those are the 5 pillars of treatment, and I'll try to discuss each of these. Now, oxygenation. Oxygen is actually a drug. Now, oxygen needs a prescription as well. I mean, we tend to sort of just give oxygen to patients, but sometimes we fail to think that it's a drug, which has to be given in a controlled way, which has to be given through the correct device. So it's an important thing. And I think this pandemic has highlighted the importance of giving oxygen correctly to patients. So the most important thing is to select the appropriate oxygen delivery option. Now, there are lots of options available: nasal cannulae, face masks, oxygen concentrators, then non-rebreathing bags, high flow nasal oxygen, non-invasive ventilation, CPAP and BiPAP. I think some of you may have seen these being used in wards. Now, generally, the nasal cannulae, face masks, and oxygen concentrators would deliver a lower level of oxygen, lower rate of oxygen, maybe 5 liters per minute, something like that. The non-rebreathing bags can deliver up to about 15 liters of oxygen. And then this new modality of treatment, high flow nasal oxygen, can deliver up to 60 liters per minute. So that's a fairly high dose. And then there are various non-invasive ventilatory strategies, CPAP and BiPAP. I think there is no time to go into details of these, but the important thing is the choice of delivery option depending on 2 things: the patient tolerance and the oxygen flow rate that is needed. Now, the oxygen flow rate is determined by the degree of hypoxia. So now, if a patient is not keeping, you may have seen these non-invasive ventilation masks, which are quite bulky, and patients do not like those. So if they don't tolerate that, we cannot simply get the patient's oxygen level up by using those. Then we may have to use things like high flow nasal oxygen, which is more comfortable for the patient. So lots of adjustments are needed. And one other important thing is proning. Now, proning is to get the patient's position changed. We get them to lie on their left lateral side, the right lateral side, and maybe even face down if they are comfortable with that. This is to recruit more alveoli into the oxygenation process. So that's an important strategy, and that has been shown in trials I think Prof. Shalini also alluded to that. That has been shown in trials to improve survival. So that's an important step. Then fluid management, that's also important. These patients, when they first come to hospital, because some of those patients do come late, and when they do come to hospital, some of them are quite dehydrated. So their fluid status has to be assessed quickly. And usually we use IV normal saline to hydrate them. Of course, sometimes we may have to give fluid boluses as well, depending on the degree of dehydration. And we may have to use other agents like IV noradrenaline if the blood pressure is found to be low. But later on, once we manage those patients, especially when they're in the ICU, we tend to be more conservative with our fluid management, because more fluid might worsen their outcomes. Then steroids, yeah. So this is something that has revolutionized the treatment of COVID-19. And there was a trial, if you may remember, which was published which was the results of which were announced in about May or June of 2020, published later on in the New England Journal of Medicine. It was the recovery trial. And the steroid arm in the recovery trial showed clear mortality benefit. And that was the first instance where a drug was found to reduce mortality in COVID-19. So now it is widely practiced globally. And we use a dose of IV or oral dexamethasone, 6 milligrams daily for 10 days or until discharge, whichever comes first. So this is in patients whose saturation has dropped below 94%. So that's an important thing, because there is a tendency to abuse steroids, and there is a tendency for patients to be given steroids even in the first few days of their illness when they are not hypoxic. And that is dangerous because that can delay the viral replication, and the patients will go on to have adverse outcomes. So it's very important that the patients are not given steroids if they are not hypoxic. So that is a point which I need to stress. Then if dexamethasone is not available, of course, it is available in most of our hospitals. Alternatively, we can use oral prednisolone 40 daily, oral methylprednisolone 32 milligrams daily, or IV hydrocortisone 50 milligrams 8 hourly. Now, IV hydrocortisone is preferred initially if the patient presents with septic shock, because it has more mineralocorticoid action. So we tend to use IV hydrocortisone in those patients, and then later on maybe convert to IV dexamethasone. And the vital thing is monitoring of blood sugar, because it's important to know that steroids will drive the sugar levels up, and that itself can be an issue in these critically ill patients. So it's important to monitor the blood sugar. In pregnant women, IV dexamethasone can be used, but it's always better to get an obstetric opinion on that. But it is used in pregnancy as well. Now, the other important thing that we need to start the patient on when their saturation level drops below 94% is anticoagulation. Because trials have shown that these patients actually do well with anticoagulation. And there have been lots of studies done about the pathophysiology of COVID-19. And in a case series of postmortems done in Italy in the early part of the pandemic, it was shown that it was a small study of about 20 patients, about 15 patients had microthrombi in the lung, and about 5 patients had actual pulmonary embolism. So coagulation coagulative defects seem to play a big part. So thrombosis is a big issue in COVID-19, unlike in other respiratory illnesses. So when the patient drops the saturation to 94% or below, we start the patient on a prophylactic dose of anticoagulation. So as for prophylaxis of venous thromboembolism, so a commonly used drug is subcutaneous enoxaparin, 40 milligrams daily, is given to all patients. Actually, when the patient presents to hospital, if the patient is ill and if you see that the saturation has dropped, then we do start them straight away. And if the creatinine clearance is less than 30 in patients, then we use a lower dose of enoxaparin, 20 milligrams a day, or we can alternatively use heparin 5,000 units 12 hourly. And in patients with end-stage renal failure, heparin 5,000 units 12 hourly is used. So the important thing is these patients can bleed, especially if the patients go on to need ventilation. They can bleed with anticoagulants, so you have to be careful. And if you, at the first sign of bleeding, you may have to stop. That is something that you have to be wary of. Then immunosuppressive treatment. Now, what recovery trial showed in another arm is that when the interleukin-6 blocker, tocilizumab, was used on top of dexamethasone 6 milligrams IV in patients with severe disease, there was a mortality benefit. So there was a clear mortality benefit, as Prof. Shalini said. So there was a clear mortality benefit. So this is in the group of patients who have severe inflammation. Now, tocilizumab cannot be used in an ad hoc manner. So in Sri Lanka, what we practice is that we start the patient on the steroid dose, and we sort of establish the patient on the steroid dose for 24 to 48 hours. And then if we see that the patient's oxygen demand is rising in spite of giving steroids, and if the oxygen saturation is not able to be maintained over 92%, and if the patient's CRP is over 75 milligram per liter, and if in those instances, we use tocilizumab provided that there is no bacterial sepsis in the patient, because it's important that tocilizumab, being an immunosuppressive treatment, can worsen bacterial sepsis if it is already there. So what we are guided by clinical evidence, cultures, and also by this parameter called procalcitonin, which is a marker which goes up with bacterial infection. So these markers are used to establish that there is no bacterial sepsis, in which case tocilizumab is used. And we use tocilizumab in an infusion, 8 milligram per kg infusion. Usual dose is about 400 milligrams given as a single dose. In exceptional cases, we do use a second dose, but that is very rarely. And usually when we think that a patient needs tocilizumab, we have a multidisciplinary meeting, because it's not a drug to be used in a lax manner. So the consultant physician, the chest physician, maybe the microbiologist, we do get together and discuss and then decide to give tocilizumab. But it's important to note that if we do think that the patient needs tocilizumab, that is in this group of patients who have severe inflammation going on, which is not suppressed by the steroid, we have to start it early as well. We cannot delay that, because then the benefit would be lost. There are some contraindications for tocilizumab as well. Active TB, herpes zoster, bacterial sepsis, I already talked about. Then GI perforation, multiple sclerosis, allergy to the drug, and if the liver enzymes are more than 5 times high. Now, COVID per se can raise the liver enzymes, but usually they are not very high. But there could be other causes for very high liver enzyme elevation, and then we cannot use tocilizumab. And also, if the platelet count is less than 50,000, or if the neutrophil count is less than 500 per cubic millimeter. So those are the instances when we cannot use tocilizumab. Then the other important aspect is the antimicrobial treatment. Now, in the setting of COVID-19, because it's a severe viral infection, secondary bacterial infection could occur, secondary bacterial or fungal infection. But in COVID-19, we are using steroids, we are using immunosuppressive therapies like tocilizumab. So the risk of secondary bacterial and fungal infection is much higher. And a particular bacteria which we are worried about is Pseudomonas, but even other gram-negative bacteria are a problem, especially if these patients, when they go on to be in ICUs for long, towards the latter part of the second week, like we do see these bacterial infections, and it's a huge problem, because sometimes patients actually do not die of the virus itself. It could be the bacterial infection that leads to death of the patient. And fungal infections, 2 fungi are important: aspergillosis, which can occur early in ICUs. Then there's a late complication called mucormycosis. You may have heard of these black fungus in India. There was an epidemic of black fungus. Those develop because the patient is immunosuppressed by steroids, then by tocilizumab or other anti-inflammatory therapies. And also, we have to note that most of the patients, some of the patients with severe disease are actually diabetic patients. So in these diabetic patients, blood sugars are also high. So all these factors favor secondary bacterial and fungal infections. So moderately severe bacterial pneumonia is generally treated initially with ceftriaxone 2 grams IV daily, plus minus azithromycin. Azithromycin is not given on a regular basis. We do give ceftriaxone 2 grams IV daily mostly. If there is severe bacterial pneumonia, of course, based on cultures as well, and with multidisciplinary input, we can give meropenem 1 gram IV 8-hourly, or piperacillin tazobactam IV 4.5 grams 8-hourly, or ticagrelin clavulanate IV 3.2 grams 8-hourly, or this high generation cephalosporin, cefepime 1 gram IV 12-hourly. But we must remember that these have to be based on cultures. Empirically, we could start, but we have to base our continued treatment based on cultures. So that's an important point. So if we have any issues with suspicion of a fungal infection, we do get microbiology opinion, and there are special tests that are done, the galactomannan for aspergillosis, and other appropriate investigations are done on the advice of the microbiologist. And one other point to remember is that there could be either co-infection of other viruses or even reactivation of viruses like cytomegalovirus, herpes zoster virus, and varicella zoster virus. So we must be on the lookout for those as well. So it's quite a challenging task managing these patients because various complications could occur. Now, again, to get back to the flow which I sort of made you follow initially. So critical disease is when now, say, most of these patients we try to manage in our ward setups. Now, if you take the National Hospital, for example, we do have designated COVID wards, and these patients are managed initially in those designated COVID wards, but then some of those patients with critical disease will go on to need what we call HDU or ICU care. So we have at National Hospital, we have 2 HDUs, one male and one female. And then there is a dedicated ICU. The medical ICU is dedicated to COVID patients. And then, of course, we do have the surgical half of the surgical ICU as well. So quite a setup is there to manage these patients. And we do see, just to sort of for your interest, we do have about 200 patients who are being treated currently in the hospital, and close to about 100 patients are actually oxygen dependent. So it's quite challenging managing these patients. And so the indication for escalation of treatment to get the patient to an HDU or ICU would be the following. If even while they are on maximum oxygen, if their oxygen saturation is less than 90%, that is with maximum oxygen support, then the patient needs HDU or ICU care. And if the patient's oxygen, even if it is above 90%, if it is less than 94%, and if the patient has evidence of increased work of breathing. Now, this is also important. The patient may maintain a saturation of 92%, but the patient may be very, very tachypneic with very high degree of work of breathing. Then there may be X-ray changes, more than 50% involving the bilateral lung fields, which indicates a very severe pneumonia. And there is this ratio called partial pressure of oxygen to FiO2. And if that is less than 300, and if there's hemodynamic instability, low systolic blood pressure, high heart rate, high lactate level in blood. So those are indications to transfer the patient to an HDU. So it's important that those patients are transferred early so that complications could be minimized. So for these critical patients, again, we can use various modalities of oxygen delivery. Now, we use these modalities even in the ward setup, actually, but even in the HDU or the ICU, we can still use them. High flow nasal oxygen, we start with a low rate of about 15 to 30 liters per minute, because oxygen is quite expensive, and actually a person on high flow nasal oxygen could consume quite a bit of oxygen, and we need sort of say hundreds of these jumbo cylinders to manage say about 100 patients per day. So it's very important that we start them on a low dose and then go up if needed. Again, the target saturation is about 92 to 96%, but of course in patients with COPD we can aim for a lower target like 88 to 92%, and must always check the oxygen stock in the hospital. I think we have heard reports of various issues with oxygen delivery in other countries, but so far I think in Sri Lanka we have been able to manage that very well, because the oxygen stocks have to be always sort of checked and replenished when we treat these patients. So then CPAP is a mode of oxygen delivery where we give oxygen at pressure. I will not go into details of these. Then we have what we call proper ventilation, non-invasive ventilation with a BiPAP machine. Here we can give what we call pressure support, or we can have different pressures for expiration and inspiration, very useful in patients with carbon dioxide retention, as in COPD patients. So in all those modalities, we can use this proning. So patients on high flow on these devices can prone. What we do is we actually get their cooperation to prone themselves actually. So and when we do that, we do see an improvement in the oxygen level. And then, of course, invasive mechanical ventilation is one of the last resorts. Now, again, it's important, if the patient's work of breathing is high with these other devices, we should not delay intubation. Because if we cannot maintain the oxygen saturation of 92%, even with 70% of oxygen given, then these patients definitely need intubation. And some of them do very well with intubation. Of course, I mean, there's this general thought that if you have to intubate the patient, that is probably a pretty poor sign. Of course, the patient is having severe disease if the patient has to be intubated, but some of them do pull through. And the other important thing that we try to achieve by ventilating is to prevent this entity called self-inflicted lung injury. When the patient is taking large tidal volumes, the patient is gasping for breath, that itself can damage the lungs. So we prevent that by intubating early. And even those intubated patients can be prone. Of course, then the staff has to get involved. Then there is this last result called the extracorporeal membrane filtration, where I mean, basically in short, you take over the work of the lung. So we have only one center that is at Karapitiya. So this is not widely used. So they could have lots of complications. I think I can't go into all the details. They could have acute kidney injury. Now, kidney problems are a particular issue in these patients, and probably some of these patients have CKD, so that may be the reason. But they do have kidney problems, and myocarditis is another issue. If we do a troponin level, the troponin level is found to be high, and in some of these patients, hyperglycemia is an issue. So we try to keep the blood sugar level between 120 and 180, and liver involvement could happen, but it's not a particular problem. And they can have CNS issues as well, like delirium, seizures. So lots of complications to attend to detect and treat early. If the patient, I mean, survives the hospital stay, I mean, if you ask me for data, generally worldwide data, if a patient is critically ill with COVID, the mortality rate is about 30%. So that's the worldwide data, even with our treatment, but probably it has gone down a little bit. And if they have to be ventilated, then the mortality goes up to about 50%. In Sri Lanka, we actually see similar figures. So we have not done badly in managing these patients. So we do see these rates, and probably we do have a population that has more comorbidities as well. So I think taking that into consideration, especially in the latter part of the pandemic, I think all our hospitals have done reasonably well. So when we decide on discharge of this patient, that is when the patient has recovered from the illness, one important thing is what to do about anticoagulation. Now, of course, if they had confirmed pulmonary embolism, then they would have been given the treatment dose of enoxaparin or whatever agent that we use. And then, of course, there is a guideline for treatment for 3 to 6 months, so that those guidelines can be followed. If we sort of gave a preventive dose of anticoagulation, then we can do one of several things. If we think that the patient's outpatient coagulatory risk is high, then we can give low molecular weight heparin as an outpatient. Of course, the patient has to come to hospital to get it, so it's a cumbersome thing, not easily practically not easy. They can be given direct oral anticoagulants like apixaban. Of course, they can be given warfarin as well, but then monitoring is a big issue. And so we select patients. Always the decision whether to initiate anticoagulation at home or not is based on two factors. We consider the risk of breathing versus the risk of thrombosis. So it's an individual patient-based decision, and we can't sort of give a general guideline on that. So there are lots of other general considerations that we have to take into account in the care of these patients. And I think I probably stressed on this fact about the optimal management of the critically ill patient in the ICU, the fluid management, the nutrition, and of course, I think Prof. Shalini alluded to this. I mean, lots of drugs are being sort of promoted as possible agents for use in COVID-19. For example, vitamin D, vitamin C, zinc, but there is actually no evidence for these. And if you followed my guideline, I think I have not included any of those because we do not give those on a routine basis to our patients. The story with vitamin D, I think, as Prof. Shani rightly said, patients who had severe COVID-19 had some of those patients had low vitamin D levels, but they also subsequently found that vitamin D levels are anyway low in patients, in diabetic patients, in obese patients, and in patients with, say, various comorbidities. So probably it was a comorbidities that led to severe disease more than the low vitamin D level. Vitamin D level may have been an innocent bystander. So we don't use vitamin D. There is not much evidence for zinc and vitamin C either. So as of now, we stick to these main modalities of treatment, and they have worked. And I think the WHO is also our guideline. The Sri Lankan guideline is mostly based on the WHO guideline, but it has taken into account other guidelines like the National Institute of Health guideline and the NICE guideline as well. Of course, one difference is that we don't use IV Remdesivir here as well. And as Prof. Shalini said, there is no mortality benefit, only a shortening of duration of hospital stay by 4 days. So because of that, the guideline groups here thought that considering the cost of that treatment as well, it was not practical to use it, and it has side effects as well. It can't be used in liver disease, in patients with liver disease, renal disease. So we do not use that. But then, so what we use is what the WHO has recommended, and we do use tocilizumab here in selected patients. Now, I can't go into details. There are special groups, pregnant patients and patients with who are immunocompromised. Their management is basically the same. We use the same drugs, no special differences. Staff safety, of course, I just brought this in as a consideration because it's important that our students, now they will go on to be house officers soon. So it's important to wear the appropriate PPE, and it's very important to get vaccinated. So that's something, I mean, if ever there is a person who thinks that vaccination is not important, I think it's very, very important that people are vaccinated. So again, the point that I want to stress is that these patients are very heterogeneous. They have different needs, and their management has to be tailored. So it is individual management, which is given based on the guidelines. So thank you very much, and I must thank SIRHA and everyone who organized this important workshop for giving me the opportunity to speak on the management of COVID-19. Thank you.
Thank you, sir. I think it's a very valuable topic to discuss, and there are so many knowledge that we can get from that. And also, I would like to go to the chat. There are some questions to ask about the management. Sir,
yeah.
What are the non-long-term complications of COVID-19, and is there any effect from COVID infection on future pregnancies?
Yeah. Well, long COVID is an important topic. Now, we discuss post-COVID illness under two categories. One is acute post-COVID illness. That is, illness up to about 3 months, so that is called acute post-COVID illness. And then beyond 3 months, that is what is truly called long COVID illness. So in the immediate post-acute period, some of these patients can have issues with breathing. I mean, a common complaint is that even when patients are discharged, when their saturation improves, they commonly complain of shortness of breath on exertion. Actually, we don't discharge these patients until their oxygen saturation has come up to at least 96%. And also, we make sure that we do that. I think I mentioned about these exertion tests, where we get the patient to take a few steps, maybe 30, 40 steps, or do that 1 minute sit and stand test. And we check their saturation to make sure that it doesn't fall when they exert. If in any patient the saturation falls, we don't discharge them at that point. We keep the patient for longer. So some of the patients do end up being with us for maybe 2-3 weeks actually. And when they are sent home, they sometimes complain of shortness of breath on exertion. They may have an ongoing cough going on for some time. Some of these patients may benefit from inhalers if they have bronchospasm. So we do prescribe inhalers to them. And of course, an important part is what we call lung rehabilitation. So in some of those severe patients, we do get them into chest clinics, into physicians' clinics for rehabilitation, where we get the help of physiotherapists to work with them, to lessen their symptoms. It's not really drug treatment. Some of these patients, I did not discuss that, but it's going to complicate the discussion. Some of these patients can develop what we call an organizing pneumonia, which is like, it's not an infectious pneumonia, it's an organizing pneumonia, something like interstitial lung disease. So it's called acute fibrinous organizing pneumonia. So when that entity is there, these patients will require steroids to be taken at home. So those patients are given steroids. Then there have been instances of patients reporting on some vague symptoms like inability to concentrate, lots of body aches, headaches going on, and sometimes even confusion. Now, say we do see what we call a post-viral syndrome in other illnesses as well. So even with COVID-19, you can see those. And it's more, I think, in COVID-19 than with other viruses. So these are other things that we have to sort of consider. The long COVID is when these symptoms persist beyond 6 months. And actually, we don't have much data on exactly what's going on. And there's a large study in the UK going on where I think thousands of patients are being studied for long-term issues with COVID. Those trials have not reported yet. And they're doing scans, they're doing lung scans, they're doing brain scans. So probably in the next few months, we will get to know more about the exact post-COVID complications that they can get.
Okay, sir. Thank you. And there is another question, sir.
Yes.
Sir, what is the reason for commencing anticoagulant therapy with saturation below 94%?
Yeah. So now when the saturation drops down to 94%, it indicates that the patient is having a severe pneumonia. So that's what it tells us. So the patient is having severe COVID illness, severe COVID pneumonia. Now we know when there is pneumonia, when there's severe illness, there is what we call, so the body has not been able to clear the virus. Now, say I told you about 80% of the patients with COVID-19 being able to clear the virus within 7 days, within 1 week. But these patients, when they go on to develop pneumonia and hypoxia, they have not been able to clear the virus. But of course, the immune system is working overtime. The immune system is trying to clear the virus. So there is what we call a pro-inflammatory state. Whenever there is a pro-inflammatory state, there's a risk of there's a high risk of thrombosis. So to prevent that risk of thrombosis, we start the patients on a preventive dose of anticoagulation. And then when we follow these patients up, at any point, if we think that the patient has clinical evidence of pulmonary embolism or clinical evidence of deep vein thrombosis, that may be based on calf tenderness, or it may be based on unusually low saturations that is not compatible with the X-ray findings of pneumonia. And of course, if their D-dimer levels are serially going up, then we know that the patient may be having pulmonary embolism or true DVT. So in which case we escalate the anticoagulant dose to a treatment dose. Otherwise, we start on a preventive dose. So the idea of starting on the preventive dose is to prevent thrombotic complications. Now, actually, I told you about this postmortem study from Italy. So almost all patients had some form of coagulatory defect. About 15 patients had microthrombi in the blood vessels of the lung. And about 5 patients had true pulmonary embolism. So, and we know that deaths could be due to this issue of thrombosis. So we do start the patients on a prophylactic dose of enoxaparin to prevent venous thromboembolism.
Okay, thanks for the answers. And after that, we can go for a small question and answer session. As the time is going so fast, so if you have any questions need to be raised from any of these resource persons, you can unmute and ask a question. And also, one of our resource persons, Professor Shavani Sri Ranganathan, just left the meeting, and I would like to thank her for that valuable knowledge about that COVID-19 medicines, which we have never had before. So it is your time to have, if you have any questions, just you can ask the question.
So I have a question. If a patient complains of any acute complications following COVID infection, how can that patient get treatment for that from that the center that he got treatments, or can he go for another OPD, or how
do you mean to say once the patient is discharged, if they come back, like, or
yes,
during the first,
yes.
Once the patient discharged, yes. So once the patient is discharged, I mean, some of the patients, I mean, they are given a diagnosis card. So for example, if we discharge a patient from the National Hospital, we give a diagnosis card. So the patient can certainly come back with a diagnosis card to the same hospital, which is what we usually advise. But of course, in an emergency, they can go to a nearby hospital, and then the patient will be taken care of and transferred back. So that facility is there. Did I answer your question, or is it something else that you asked?
Yes, sir.
Do you have a particular issue in mind? Yeah.
One of my friend got COVID, infected with COVID, and she ended up with persistent coughing.
Okay.
So,
yes. Yeah, certainly. Yeah. I mean, if you, as I told you, sometimes a persistent cough can be there, maybe due to bronchospasm, in which case the prescription of a simple inhaler steroid inhaler might do the trick. But of course, it may also be due to something more serious, like what we call an organizing pneumonia, which can develop late. So in which case, of course, we have to admit the patient to a hospital and then investigate and treat. So if any patient after discharge is having any issues, they should report back to that particular hospital, or at least report back to a doctor, and then they will most probably go through the patient and make the necessary referral.
Thank you, sir.
Okay.
And there is another question through the chat to Professor Inoka. Madam, what is the rationale in advice given that not to bathe or wash the vaccinated site?
No rationale that I can think of. This it's just a common practice in Sri Lanka. I think it's just a cultural thing that we have. There's no rationale for giving that advice.
Okay, Thank you. If there are any other questions, you can ask by unmuting. Okay, since there are no further questions have been shown in the chat or the mute, so I would like to conclude this session. So before that, today I think we have had a vast kind of knowledge which should be have as a medical students in our career, and also it is very important to treat and work in the wards to the betterment of the patient in the wards. So I would like to thank Professor Sharan Sri Ranganathan, Professor Inuka Korea, and Dr. Harsha Satish Chandra for joining with us to this valuable discussion and having their valuable knowledge sharing with us. And also, further thanks, special thanks goes to Professor Vajra Isanayaka, the Dean of the Faculty of Medicine, Colombo, and Mr. Bandara, the Deputy Registrar of Faculty of Medicine, Colombo, and Dr. Roshan Hewapathirana, Dr. Sachit Aperatna, the Senior Treasurer of the SIRHA, and also all the members of the Advisory Committee of the SIRHA for making this effort so successful. And I would like to conclude this session now. So I thank all of you for participating in this session and I think you had a great time and a valuable time with us, and hope you can have furthermore sessions like this in the future. And I would like to conclude this now. Good night for all of you. Thank you.
Yeah, thank you.
Thank you, sir.
Thank you.
Thank you.